Hi everyone, and thanks for joining us for the third webinar in our series on development and validation updates for our open source implantable neuromodulation system. My name is Sahar, Center Director of CARSS, which stands for Center for Autonomic Recording and Stimulation Systems. I lead the operation and technology integration of our open source platform called OpenNerve. So a little bit about who we are. We are a center based at USC that was formed 3 years ago with a $12 million grant from NIH Spark program. And actually since then, we received additional funds to not just develop a platform, but also conduct in vivo studies. The problem we're trying to solve in a nutshell is to reduce barrier to entry for novel bioelectronic medicine applications by developing a platform for neuromodulation and making it open source to the public. So firstly, what's the platform that we're developing? It's essentially hardware, software, firmware, and all the tools needed for a closed-loop implantable system. At a high level, it includes an implantable pulse generator or an IPG, leads and stimulation electrodes and sensors for end organ sensing for closed-loop modulation, and patient-clinician remote to interface with these electrodes. And all of the design files and test protocols are documented and are being released on GitHub and online repositories under a very permissive license. I should note here that although we're conducting several animal studies, we're making the design open source and leaving specific use cases up to the user, which means they need to obtain their own IDE for that use case. So we've structured the platform development around 5 nested elements, which we call nests. Each nest focuses on developing a specific subset of components. Before diving into who's working on what, I want to clarify that these nests fall into 2 categories. We have the established components, which are developed under a quality management system. And then on the right side, we have our novel components, which are still in feasibility testing stage. Nest 1 through 3 are our established components. So these cover the IPG mechanical housing and a comprehensive set of sensors and stimulation electrodes like sacral nerve stimulation, PDMS cuff, ECG, EMG, and accelerometer leads. These are all developed by MedAlly. The electronics and firmware for the IPG are under Nest 2, and they're being developed by Medipace. On the other side, our novel components fall under Nest 4 and 5. Nest 4 includes a microfabricated cuff electrode designed for small branch nerve fibers under 1 millimeter in diameter. This is being developed by Dr. Alice Meng's team. Nest 5 includes a temperature sensor to monitor inflammation and a strain sensor to detect changes in inflation and deflation of an organ. And both of these are developed by Dr. Hengbo Zhao. And finally, we have a pH and chemical sensor. The pH sensor can help detect disease biomarkers like stomach acid. And the chemical sensors are designed to be implanted at nerve endings. They will target neurotransmitters such as acetylcholine and catecholamine. These are 2 key players in parasympathetic and sympathetic branches of the autonomic nervous system. So all of these components, the development of them are complete, and we're now running accelerated aging studies. So now let's talk about why open source neuromodulation platforms matter. As a lot of us know, developing neuromodulation devices from scratch is expensive, slow, and risky. A lot of researchers spend months or even years just developing the basic tools they need before they can even test their real ideas. And even if they get promising results, most projects hit what's called the valley of death. That's the critical phase between early research and clinical translation. At that point, there's no funding, no infrastructure, and no clear path forward. As a result, a lot of good science never reaches patients. With OpenNerve, we've built this plug-and-play platform specifically for bioelectronic medicine applications. Researchers can quickly get the tools they need and test the new ideas without having to reinvent the wheel. In short, OpenNerve can shorten development time and give more therapies a real shot at reaching the people who need them. But in reality, an open source implantable medical device is actually a very novel and quite complex idea. Most of you, when you think about open source, you usually think of software, where you can copy a repo, make a few edits, and deploy instantly. But when it comes to implantable medical devices, it's not that simple. You can't just copy a design and start testing it in human. These devices are subject to very strict safety and regulatory requirements that make a replication and deployment a lot more complex. But what OpenNerve does is it provides a more streamlined path towards first-in-human studies by lowering early development barriers. How? By giving you tools to test your novel neuromodulation concepts early in development, giving you a chance to optimize your parameters in large animal models, and enabling you to screen out non-viable ideas before investing substantial time and money. And lastly, you can use our upcoming quality and regulatory documentation as template, so you're not starting from zero when you're ready to file for a new indication. Another way of looking at it is looking at your options without OpenNerve. Well, you could try to use a commercial system off-label, but that's not customizable. Or you could build your own system from scratch. Again, that's expensive, takes years, and introduces delays and technical hurdles that slows everything down. So hopefully by now you understand why an open source neuromodulation system matters and how it can be used. So now that I've discussed our platform and motivation behind open source, I want to switch attention to providing a bit more detail about each component and then briefly touch base on the in vivo studies we're conducting. The intention behind these studies, I should say, is to help demonstrate capabilities and generate data for future users. So we're collaborating with several experts in specific indication and animal labs to evaluate our devices across multiple studies. And here's a summary. We have rat studies at USC focused on safety and functionality of our novel devices. The acute studies are completed and we're now running 30-day chronic studies. And then for our established components, we're conducting pig studies at Cal Poly. These are similar to our rat studies, but with more than 60-day implantation. And then we have, for Cal Poly, the first set of chronic studies was completed, and now we're planning the second cohort of studies. We also have multiple indication-specific experiments. We have one is low-frequency nerve block. This is work based on promising work from Ken Yoshida's lab, and this approach uses DC-like sine waves to block action potential propagations without damaging the nerve or the electrode. We're currently optimizing parameters in rat studies before moving to chronic pig studies. And then we have bladder control after spinal cord injury. This is, for here, we're using our cuff electrode to trigger sacral anterior root stimulation for voiding. And this is a collaboration with Lundquist, USC Keck, and Ekate. They're all experts in bladder control. And we also have a chronic constipation study. This is sacral nerve stimulation to induce colonic contraction in patients with chronic constipation. This study is in collaboration with UCLA pig lab and is already underway. And then we have celiac vagus nerve stimulation for hypoglycemia. This is we're trying to investigate vagus nerve stimulation to increase blood glucose. It's in collaboration with the University of Michigan and Reshape, and we'll begin these studies very soon. Lastly, we have dysphagia project. This is aimed at patients with stroke, epilepsy, and Parkinson's who have trouble swallowing. This is in collaboration with Rutin, and studies are done at Lundquist Animal Lab. These studies have already started, and we're refining surgical techniques currently. So there's a lot of studies going on, and these studies are key to refining our technologies and then eventually translating them into clinical applications. I want to walk you through the standard leads developed by MedAlly that we tested at Cal Poly Animal Lab first. We have a bipolar cuff electrode. It comes in 3 sizes designed to fit nerves that are 1 millimeter, 2 millimeter, or 3 millimeter in diameter. We also have an EMG lead that includes a suture for anchoring. Then there's a linear 4 electrode array, which is suitable for sacral nerve or spinal stimulation. This one also has suture anchors for securing to surrounding fascia. For cardiac monitoring, we've got an ECG lead that can be implanted in the suprasternal region. And finally, there's the AMS lead. That's our accelerometer motion sensor lead. It detects organ movement, generates digital data, and then communicates with the IPG using I2C protocol. So here's a real photo of the IPG. We have header pins, so you can connect up to 2 bipolar stimulation channels. You can also use them as 4 monopolar stimulation channels, depending on your setup. On the sensing side, it includes a 4-channel analog front end for bipolar biosignal recording, which makes it really modular. You can swap in the sensors you need and then interface them with the IPG hardware. There's also a digital sensing channel using I2C, which works great for AMS sensor. And the Bluetooth antenna is built right into the header, so it can wirelessly communicate with an external app. So we implanted leads in Yorkshire pigs for over 2 months. Before euthanasia, we performed acute stimulation to evaluate functionality, and we're now in the process of analyzing both the functional data and histology. One challenge we encountered was that the pigs more than doubled in size during the study, which led to some leads becoming dislodged. Based on that, we're planning to revise our implant strategy to better accommodate growth over time. Looking ahead, we're planning a chronic implantation study for January 2026. This will involve 6 pigs, 3 with vagus nerve stimulation and 3 with sacral nerve stimulation. We'll be running closed-loop stimulation and sensing in this cohort, and we'll assess 60-day safety post-implantation through detailed histology. Next, I want to highlight our novel cuff electrode. The electrode is fabricated using thin film technology, so the team has also developed a custom implantation tool to support precise placement on the nerve. The cuff itself is made from Parylene C with platinum contact pads and is designed to fit nerves in the 0.5 to 1 millimeter diameter ranges. It connects to a standard lead that interfaces with the IPG, and the entire assembly is over-molded in silicone for installation. That over-mold also provides a convenient way to anchor the electrode to surrounding fascia for added stability. To validate the design, both in terms of functionality and ease of implantation, we've been testing it on the rat sciatic nerve, which is about 1 millimeter in diameter. It's a good initial model to demonstrate proof of concept before scaling up to larger animals. Here you can see a successful implanted device along with recorded electrophysiology data. This confirms that the electrode can be securely deployed on the nerve and is capable of delivering stimulation. We've now entered the chronic testing phase, and then at the end of the study, we'll harvest the nerve and perform H&E staining to assess the inflammatory response or any tissue reactions. This is the first of many more animal data that we'll release on Spark DRC. Check it out for detailed instructions and feel free to reach out to discuss more. Also, stay tuned for future design of an implantation tool to deploy this electrode laparoscopically in pigs. Next is an enzyme-free electrochemical sensor with the goal to detect up or down regulation of sympathetic and parasympathetic activities, such as norepinephrine and acetylcholine. This sensor is initially tested in a substitute organ, specifically the rat cortex, to validate its ability to detect neurotransmitters. Then we would move to an organs such as the distal colon. In this study, we electrically stimulated the brain while recording the sensor's response. Also, we injected dopamine locally and observed a measurable signal. The results shown here provide preliminary evidence that the sensor can detect neurotransmitter fluctuations in the cortex subdurally. Our next step is to reduce the electrode's form factor for chronic implantation, which would then allow us to study neurotransmitter dynamics without external stimulation and assess long-term performance after foreign body response. The last sensor I'll highlight is our strain sensor, which operates on a capacitive principle. So when the sensor stretches, its capacitance increases. The sensor can be designed differently based on the size of the target organ. For example, it is designed to be longer for pig bladder and shorter to fit around rat bladder. To test this in vivo, we ran urodynamic experiments where we controlled the bladder inflation and deflation using a catheter. The sensor successfully tracked volume changes through these cycles, confirming its sensitivity and reliability. We're now moving into chronic studies where we allow the bladder to fill and empty naturally. And if you'd like to learn more about the strain sensor, check out this new publication. It highlights a study done on isolated pig bladder and shows it can follow inflation and deflation of the bladder. Moving on to our indication-specific studies that I will briefly go over in this one slide. One of our key efforts is focused on developing a closed-loop sacral nerve stimulation system to help individuals with spinal cord injury urinate when their bladder is full. In this study, we're using a clinical-grade sensor, which is a flattened PDMS cuff, to measure impedance across multiple frequencies. Through several acute studies, both in natural filling and controlled urodynamic conditions, we've consistently seen that impedance changes track bladder inflation very reliably. There are a lot of technical details that I'm skipping due to time, but the next steps are already underway. We're beginning chronic implantation studies in rats and have launched a series of closed-loop acute studies in sheep using sacral nerve stimulation electrode. Next, I want to briefly touch on the nerve block study I mentioned earlier. The concept here is to selectively block action potentials from traveling in one direction using low-frequency stimulation. This has been one of the more challenging protocols to optimize, but in our current setup, where we stimulate the nerve proximally and apply the block distally near the muscle. These are done in rat sciatic nerve. We're seeing a clear reduction in muscle activity when the block is turned on. It's a good step forward in demonstrating directional control using low frequency nerve block. So yes, a lot of studies and data in motion, but the big takeaway here is that the platform is working. And our hope is that one of these indication-specific examples can eventually serve as a template or a starting point for your own novel applications. I'll just end by saying that we're certainly not claiming we've solved everything when it comes to open source implantable neuromodulation systems, but we have taken some important first steps and we're really trying to raise awareness both about potential benefits and the limitations so this can be part of the future landscape.
Now I'll give some updates on our IPG development and how we're moving forward, as well as go into some of our open source resources and what you can find online from OpenNerve. Our IPG's form factor is designed to be familiar and to mimic what's already out there in commercial IPGs. However, thanks to MedAlly, we have been iterating and improving on the manufacturability. We've tried a number of different manufacturing methods to get better hermeticity, better yield, as well as lower cost for low volumes. And right now we're actually doing a hydroforming process. Thanks to that, we've been able to successfully manufacture hermetic devices, and these devices will be used in our upcoming chronic animal studies. So we're also doing some innovations in the actual enclosure design. One of the things we're doing, or one of the things MedAlly is doing, is machining out the titanium just above the wireless coil. The idea being that this will attenuate the signal less and allow for more efficient charging with less heating. That's something we're excited to introduce and hopefully will improve the efficiency and improve the safety of these IPGs. Another innovation we're introducing is just sort of having overlapping edges on the walls of the IPG. This will help for improved manufacturing and improved quality control in creating that hermetic seal, that hermetic weld around the can. Inside the IPG, we've actually been developing a lot on the PCB itself. So we've used the Gen 1.0 PCB in our past animal studies that Zahar described. This PCB already has a lot of features that we think that the community will like and that will help to help them develop different closed-loop therapies. This includes 8 different stimulation channels, 4 ports for analog front ends, including for ECG and EMG, an I2C header in the, or an I2C port in the header itself to allow in-body digital communication or to communicate with sensors on the, with digital sensors that are on different end organs, as well as having a BLE, a Bluetooth Low Energy antenna in the header so that it can communicate with devices outside the body, such as a continuous glucose monitor or a tablet for control. For Gen 1.5, this board is now in production. And for this, we've actually increased the AFE channels. So now there are things like AFE channels for neural signals for ECAPs. However, we have decreased the stimulation channels down to 4. One sort of thing we've added for all of the stimulation channels is a sine wave generator. This will allow us to output a low frequency sine wave that we can use for directional nerve blocking. So we can output a sine wave to block signals in one direction and then stimulate in the opposite direction. This Gen 1.5 PCB also reduces the part count and increases the efficiency and the manufacturability of the PCB, for example, by almost completely removing high-density interconnects, sorry. And this will allow it to be produced at a much cheaper price point, as well as make it easier for external groups to produce this PCB if they need to. Here are the specs for our IPG. This is for the Gen 1.5 PCB. As you can see, there's 8 channels for analog front ends, as well as still the I2C port and stimulation ports. For stimulation output, we're still rating it at 5 milliamps for our maximum output volume, or amplitude, sorry. For pulse width, we're doing up to 1 millisecond, and then for frequency, we're looking at low frequency stem up to 100 hertz. If you're interested in pushing these boundaries a little bit, please get in contact with us. There are modifications that we could make to extend this if needed. In addition, we have all of these analog front ends on here. There's analog front ends available for measuring heart rate variability or for extracting respiratory rate from an ECG signal, as well as measuring EMG from different muscles and measuring neural signals or ECAPs. All of these analog front ends can be adjusted using just by swapping resistors and capacitors on the board. So if you are using a bare PCB, it is totally possible to modify these to meet your particular research needs. And speaking of using a bare board, for all of our PCBs, we are including a frame around them when they're first manufactured to have USB power, to include a Bluetooth antenna so that it can communicate wirelessly, to include easy access to all the different channels, as well as pins for debugging and for flashing the firmware. So we hope that these can be used as development boards, and in fact, we already have users that are using these. And going forward as well, we're going to try to reserve several PCBs in each manufacturing run for development boards for people who have very early stage research and want to try the OpenNerve platform early. All right, now I'm going to talk a little bit about sort of the resources that we put online, as well as the future plans for our center. Our center's goal is to develop everything that we do under an open source protocol. So what that means is that as different things are developed and tested, as data is collected, as source files are generated, all of that goes online on our GitHub and are released under an open source permissive license, which I'll explain in a second. We already have a lot of files online, and we're continuing to add files again as, for example, the new PCB gets manufactured. But right now, this is a snapshot of what's online right now. So for our IPG, we have basically the complete files for our Gen 1.0 IPG. This includes the schematics and layouts for the PCB inside in Altium, as well as PDFs so that you can look and see the schematics or look at the different layers. This also includes the firmware, as well as a very early version of Windows control software to communicate via Bluetooth and to turn on and off stimulation, adjust the parameters. We're also including things like design documents, design inputs, testing documents, and testing protocols. So we hope that those can be useful even for users that aren't necessarily using our IPG in order to guide the development of their own implant. Similarly, for our standard leads, this would be for our silicone cuff, our linear array mimicking an SNS array, our ECG and EMG electrodes, as well as our AMS electrode, the motion sensor. All of the design diagrams, all of the mechanical drawings, as well as CAD models are available online. We also have a benchtop testing plan for those leads. It should be worth noting that the IPG and the leads, that testing is ongoing, and as soon as that testing is done, all the data will be released on our GitHub as well. Similarly, for the devices developed at USC, we're releasing everything as it's developed. You can see that this includes from our Paraline cuff, which has CAD files, things like our microfabrication masks and the microfabrication protocol to actually build these and assemble them, as well as a lot of really interesting documentation about the design selection and how we came up with this design in the first place. For chemical sensors, right now we have up our masks for microfabrication, as well as the fabrication protocols. And then for our mechanical sensors, for the strain sensor and the temperature sensor, there are fabrication masks. We included CAD models there for the testing setup, so people can validate these themselves if they'd like, as well as some initial characterization results. And more of that is coming out as those studies are being finished. All of the design files that we release are being released under this Creative Commons CC BY 4.0 open source license. Now, this is a very permissive license. This is not a completely permissive license. We're not releasing these under the MIT license. But the Creative Commons license allows anybody to basically use our material for anything as long as they give us proper credit, which can be as simple as citing one of our papers. Now, what this license means is that if you're an academic user, you're welcome to look at OpenNerve's documentation and use it whatever resources you can. If you're a commercial user, if you're even a large company, but also especially small startups or people trying to get a head start, you are welcome to use what OpenNerve has created in order to get your product to clinic faster. One thing we really wanted to do was enable people to raise money and to fund these clinical trials that'll actually get an implantable device to a point where it can help people. And so we thought a permissive license was the best way to go because it won't limit any of the IP generated by a commercial entity. Of course, we really want to continue working with people. We hope that users will continue to make things open source or will take any innovation, any improvement they make and contribute it back to the OpenNerve project. But we didn't want to limit anybody or cause any sort of fear or apprehension because of a share-alike clause, for example. If anybody has any confusion or any questions about our open source license, I encourage you to ask them after this or reach out to me directly. I would love to talk to you about exactly what your needs are and really what you think about open source medical devices in general. So there are a number of ways that users can work with the CARSS Center. The users can use OpenNerve for their research. So as I just mentioned, our files are all online and available. Any of you are welcome to take a look at it and to get any inspiration or to use these files as needed to improve your work. However, we're also more than happy to work with you more directly, whether providing hardware, providing devices, collaborating on a grant, collaborating in another way, co-developing things. We're very open and available for all of those options. Some ways that people have worked with us or have expressed interest in working with us include, on the very early side, getting these development boards. So getting PCBs with the frame included that they can use for animal studies, as well as getting some standard leads or even some research leads that they can use in percutaneous or subchronic or even chronic animal studies where the leads are externalized and can be interfaced with an external stimulator. So we've done that before, we're happy to do it more often. On the chronic side, sort of moving forward, we have, thanks to MedAlly and their expertise, multiple options for chronic implants. It's absolutely possible to get a hermetic sealed sort of human level enclosure on OpenNerve and to use that for your animal studies. However, we're also happy to explore options like replacing that rechargeable battery with a primary cell, or using a non-hermetic polymer enclosure, something that might be a little bit cheaper or a little bit easier to manufacture. We're very open to all of that, as well as if there's any modifications you need to make or any sort of small changes that you need to make, but maybe your research lab doesn't have the capabilities, reach out to us, let us know, and we can see how we can help. And then of course on the clinical side, MedAlly is an ISO 13485 manufacturer based here in the United States and would be more than happy to work with you and support your clinical trials using OpenNerve. So why use OpenNerve for early stage research? I think there's a lot of advantages. I hope that there's a lot of advantages to using our technology early that will hopefully make things easier as a therapy progresses from sort of early percutaneous studies all the way to the clinic. So on the early side, if you use OpenNerve, or our vision at least for using OpenNerve for small exploratory trials, is that it will then reduce the cost and reduce the risk of moving to that next stage of chronic implants. If you use OpenNerve for a rodent study or for an acute animal study, you don't have to redesign a device or design a new device from scratch versus if you were, say, using a National Instruments board where you wouldn't be able to implant that, you would have to create your own device. You can validate if you're using OpenNerve, you can validate your therapy using the actual OpenNerve hardware on the benchtop and then quickly move to that next stage of development. Also moving forward, if you're using OpenNerve, this is a device that, again, can be manufactured under the standards necessary for use in the clinic. And so using something in chronic animals, there's not a need to develop a new device after that to go to a clinical trial. Unlike if, say, you were using a more researchy device and you needed to make something that could be manufacturable in a QMS facility. Or if you were using a device from somebody like Medtronic and you would have to sort of raise money to create your own device if you wanted to move that forward to a therapy without them. Having said that, OpenNerve is still in the early stages. We haven't gotten to a clinical trial yet. We're still working on our animal studies, as Zahar mentioned previously, and we're looking to continue those and finalize all of them within the next 6 to 9 months, as well as bringing on new collaborators, new partners in order to continue that sort of early to middle level research in pushing toward new therapies. We are always looking for people that are interested in taking OpenNerve to the clinic, especially people that are interested in taking it to the clinic and then releasing some of that documentation as open source. What I mean by that is not necessarily releasing clinical data. That is something that we're actually going to stay away from. But I think that there's a lot of value to the community in, say, having an IDE application that is open and available as sort of a template or for people to learn from, or having FDA communication, meeting minutes for a Q-sub. Those are things that I personally would love to see OpenNerve be able to release. And so if anybody listening or if anybody on this call is interested at all in potentially doing an open source clinical trial, or like doing a clinical trial in partnership with us and releasing some of that sort of the protocols and the templates to the community, please let me know. I'd love to talk to you. In terms of sustainability moving forward and bringing on board new projects, we're actually very lucky that there are two sort of established commercial entities that have agreed to take on this, this project and continue sustaining it throughout the future. So MedAlly, of course, is committed to manufacturing OpenNerve, as well as committed to supporting it in the future. They're actually developing a stock of components, a stock of IPGs and a stock of leads that can be shipped out sort of within the week. So this is a huge vote of confidence from them, and I'm excited that they'll continue to work with customers to get OpenNerve and to make sure OpenNerve stays sustainable. And similarly, Medipace is committed to continue manufacturing these PCBs, both for MedAlly as well as dev boards for different people that might need them for early stage studies. And on USC's side, so USC has actually just received a POSE Phase 1 grant from the NSF. This will allow us to do things like look deeper into what a clinical trial with open source documentation release would look like, to look into what sort of governance structure might exist for the open source files, and more importantly, or most importantly, to look into how to form a real community around our open source project. Right? We are more than happy to have developed these resources from the community, but what would be even better is to get people from the community to pour back into the project, and for more people to sort of continue improving OpenNerve, adding new features, adding new leads that can interface with our IPG, and just sort of expanding it and making it even more useful for more and more people developing therapies. So that is going to be our goal over the next year, to get this sort of creator community rolling. And on that note, if you are an open source developer, or if you're somebody that develops medical devices and is at all interested in open source, please reach out. I would love to talk to you, and I'd love to see how we can continue developing this in a way that benefits you and brings you sort of onto our team. Because I think that's one of the big promises of open source, is that it can become this a lot bigger than one group developing and can actually become a community. So that's what I've got. After the recorded presentation, I am going to sort of take a few minutes to show you around our GitHub to display some of the files we have online right now. But before I do that, I'd love to just give a shout out to all the great people that are actually making all these things, and to just say thank you to everybody that's been involved in the project. This includes, of course, all the three groups at USC. This includes Medipace and MedAlly and the great people working there. Our external advisory committee, thank you guys so much for the advice over the years. And of course the NIH, Eric Hudak and Brooks Gross, who have both believed enough in open source technology to fund this in our sister project, as well as continued to support us throughout the last several years of development. So thank you guys. And of course, please reach out and get in touch. Feel free to email us at cars@usc.edu. Go to our GitHub, like I mentioned, all our resources are online and available. And then our website we'll have some periodic updates on there. We also will post the recording online on our website. So if you're interested in watching this again, please go to our website and check it out. Thank you very much.